So untreated plasmid gives a number of bands depending how they are uh, positioned into the gel compared to the treated plasmid with a restriction enzymes. So as we have seen in here in this gel, that's the untreated plasmid gives a nicked band. The same plasmid gives a nicked band, sometimes it's a linear band, but mostly you will see the supercoil band. This is the most abundant form of band it will be detecting in a untreated plasmid. And for the treated plasmid, if the restriction enzyme cut the plasmid at one site, it's just linearize the plasmid. But if the restriction enzymes cleaving the plasmid in a multiple sites, then you will get a multiple bands into the onto the gels. So the supercoil is the native form conformation of a plasmid in host cells, and it is the most occurring uh, form of plasmid DNA. On the other hand, like during the replications, the cellular topoisomerism, which can nick one strand of a double-stranded plasmid, just nick a one strand, and that gives you a nicked plasmid band. Again, so the plasmid has been extracted using a alkaline lysis process. So normally the bonds, as we have seen, that the bonds, the bases of the double-stranded DNA are held together by a hydrogen bond. So during that alkaline lysis conditions, those double-stranded hydrogen bonds are disrupted, and they are actually then denatured to have a single-stranded bond. But when this alkaline pH has been uh, coming back to the neutral conditions, these hydrogen bonds can form together, and they are producing the supercoil one. That's the most abundant one. But if that alkaline lysis process is too harsh or has been done more than the required time, then they cannot come back, or the hydrogen bonds cannot be reformed again. That means that the DNA has been denatured uh, permanently, and that sometimes gives you a single-stranded plasmid band onto the gel. And again, with the treated one, you always get a linear form of a uh, plasmid, but you might get some supercoil as well, depending on the concentration of the plasmid and the restriction enzyme, how much restriction enzyme actually has been used to check uh, the digestions. Or so it is not very important. It's mostly important that whether the plasmid has been cut in one places and you actually got a strong band of a linear strong band of a linear plasmid. That's the most important bit. So restriction mapping is a process that has been used again with the restriction enzyme, and it is mostly used to actually identify the unknown DNA fragment. So you got a DNA fragment, you don't know any of the characteristics or the sequence of this DNA fragment. You just try to using different kinds of restriction enzyme and try to map them from that uh, digestion. So in these examples, we can see that the plasmid has been digested with a EcoR1 and BAMH1, and also both EcoR1 and BAMH1. So the EcoR1 has been giving a three size bands, that is 1.9 kilobase pairs and the 1.4 and 2.2. On the other hand, the BAMH1 giving another three bands of like a 3 kb, 1.5 and 1 kb bands. But when they are digested together, you can get actually 1, 2, 3, 4, 5 fragments. And these 5 fragments then in here, it is showing that you actually map them, that what might be the size. So this is the BAM H1 3 kb band, and this 3 kb band actually cutting by one of the Eco R1 site, which gives a 1.9 and 1.1 kb bands. And then this 1.5 kb band, which has been cutting by the BAM H1, is 1.5 kb, which has been cutting by another Eco R1 site, which is give a 0.3 kb and 1.2 kb. And this 1 kb band is here. So you can actually make the whole mapping of some unknown DNA fragments with the restriction mapping. Also, you can use this restriction mapping to confirm your DNA sequence in a plasmid. So it can be used these techniques for molecular cloning, as well as to get information about unknown DNA fragments. And this has been used routinely into the labs to check the molecular clones or the unknown DNA fragments. And the other process, as we have said, that the Southern blotting, which actually used to determine the number of copies present into the host genome host organism. So in this process, so normally when you get the plasmid of expected DNA, so you want to put that DNA fragment into an host organism, like yeast. That's a Saccharomyces cerevisiae, which is most abundantly studied organism throughout the world. So you want to transform that specific plasmid with a specific DNA sequence into the yeast. So in this case, you want that one copy of that gene has been integrated into the yeast genome, rather than getting a multiple copies. You can get a multiple copies depending on the downstream processing. So like if you want to check that a gene has been expressed in a yeast or not, that expressed means whether it is produced the protein or not. So you might have a multiple copies, but in some specific cases of experiments, like sometimes using in a CRISPR, where you actually want a one copy of that gene, like a Cas9 protein. The Cas9 protein has been required to do the CRISPR mechanism, but you do not want the Cas9 protein to be expressed more than the required. So you want just one copy of that Cas9 protein into the genome of the host organism, maybe yeast, maybe fungi, maybe a mammalian cell line, whatever you are choosing for your experiments. So in that case, we need to identify that the host organism actually having just a one copy of that gene, not a multiple copy. So if you have the multiple copy, you can get a more mutation into the genes. But if you have a one copy, you have the mutation, which is actually you are expecting to. So in the Southern blotting, what has been done that you actually transform your plasmid with the expected gene, and then you actually extract that genomic DNA from that organism. For example, if you say that you actually transform the plasmid to the yeast, so after selecting the yeast, either from antibiotic resistance or growing in a specific nutrient, you select the transformant. And from the transformant, you grow them, and then you extract the genomic DNA. That genomic DNA has been actually digested with a restriction enzyme. So you need to know which restriction enzyme you will use. So I'm just telling the general process. So that genomic DNA has been digested with the restriction enzyme. And then this digested product has been run into the agarose gel. So after running into the agarose gel, this bands has been transferred to a nylon membrane. Mostly is a nylon membrane, the genomic DNA. After a transformation, nitrocellulose membrane, not the nylon membrane, sorry about that, nitrocellulose membrane. And then this nitrocellulose membrane has been fixed with a UV radiation, and then it has been hybridized with the probe. So we have been used the probe, if you can recall, we have used the probes in a DNA hybridization experiment. So something like that, we have to use a specific probe of that specific sequence and has been treated the membrane with that probe. So if you have a 1 copy or 2 copy or 3 copy, depending on how many copies of that specific gene has been integrated into the genome, you actually get a multiple bands. So if there is just a 1 copy, you might get just a 1 band or 2 bands. But if you have more than 1 copy, then definitely you will get a multiple bands in a maybe 3 or 4 bands. So from that information, you can tell how many copies are actually integrated into the genome. So this is the process of Southern blotting, and its most important application is to determine the number of copy of a gene of interest in a host genome.